All right. Thank you, everyone, for joining us. It's my great pleasure to introduce Professor Seth Bordenstein from Penn State University. Uh, just wanted to share a little bit of background about Seth. He completed his bachelor's, master's, and PhD from the University of Rochester, and then started his lab and career at Vanderbilt University, where he built a really vibrant community of researchers studying the microbiome and has been really focused on looking at animal microbe symbioses and has extensively studied Wolbachia bacteria and bacteriophage in flies, mosquitoes, and wasps. Recently, in 2022, Professor Bordenstein moved to Penn State University, where he's both endowed chair and also directs the Penn State Microbiome Center. And just by tracking how things are going with the center, it looks really awesome. More than 500 people are part of this center, and there's some really exciting research coming out of it. And not only that, besides Seth's really illustrious research career, he's also been deeply engaged in education and has been leading an HHMI initiative on Discover the Microbes Within. So it's my great pleasure to introduce Seth, Professor Bordenstein, and I'm really excited to hear his talk. So I'll hand it over to Seth.
Excellent. Thank you very much, Danita, for that introduction. It's an honor to be invited by you and your group to give a talk. I'm sorry that I can't visit. My schedule as a director of the center is a little silly these days. And to get out in this time of the season, we had to do a Zoom, but I will definitely come back in the future if you have a microbiome symposium event or something like that. And I think we'll have a Q&A session open to everybody after this talk. So please hang out if you'd like to
know
a bit more.
If you want to stick around and hear more about Seth and get to know him, I thank you, Seth, for reminding us.
Yeah.
Hang out where the room will be open.
Yep. Total just hang out and ask me anything. Okay, so I'd like to tell you a little bit about my journey to where I'm located right now, and then also our journey through the science. So let's go ahead and begin. I know you have a vibrant microbiome community at UCLA, and you're all very lucky to have Nandita as a member of your faculty. What I would like to tell you about is what we're doing just in a couple slides at Penn State and why I moved here. We are really forming a nexus center between these two very large academic and industry spheres. The first is, many of us are aware that there's a Microbiome Centers Consortium. This is about 80 places worldwide that have an academic unit dedicated to microbiome sciences. And there are about 100 One Health academic units throughout the world as well, where the emphasis of study is typically on flow of microbes and pathogens through various ecosystems, including animals, humans, and the environment. Now, we recently named our center the One Health Microbiome Center as we reflected on a lot of our expertise at a land-grant institution like Penn State. We have a deep, rich set of labs, in fact, 125 labs affiliated with our microbiome center because they represent some fundamental ecosystem aspect of human health, environmental health, and more broadly, agricultural health, because we include plants and animals and ag all in this. And of course, microbes are the base of the biosphere, no surprise to any of us today, but those microbes determine the outcomes of health and disease in each of those ecosystems for each of these types of hosts, and those microbes may flow through those ecosystems that represent the pillars of a One Health paradigm. So our aspiration and really our belief in what the Penn State Microbiome Center offers is this kind of non-specialized, multi-scale, multi-ecosystem set of knowledge and applications driven by microbial wonder and discoveries. In that context, I'll just give you a few numbers. So we have 540 members, 125 faculty from 42 departments. Penn State is a very large university and can gather that kind of membership. There's 160 graduate students. Some of you may be thinking about graduate school or advanced professional degrees. In fact, we are offering the first microbiome sciences PhD degree in the world, as far as we know here, right through the One Health Microbiome Center. This is offered as a dual title degree, so you get to maintain your core programmatic study and then add on the microbiome sciences PhD title to affirm and accredit the training environment and training of our students. We have a biotech science education series that Nandita referred to called the Wolbachia Project, which is an insect microbe biodiversity study that includes biotechnology, bioinformatics, and a lot of biotech at the high school and intro to college level that's spread across the world. And this is under the framework of our Discover the Microbes Within program. So for anybody that wants to hear more about it, we got websites and socials up. We're active on all the socials, including YouTube. And we have any interest for graduate studies, check out our graduate programs at this Huck link here. Okay, so without further ado, let's go ahead into the areas of research that our lab is focused on. We've been studying symbiosis in microbes in animals for 25 years now. When Nandita invited me to give a talk, I asked, well, we got to figure out which one we're going to talk about, or we'll talk about all of them today at a very light level. And so Nandita suggested I talk about microbiome variation within humans. And really that comes out of my interest in thinking about what are the rules of microbiome variation between species, where we focus a lot on the determinants of how phylogenetic history of animal and plant hosts influence microbiome assembly through phylosymbiosis, but also what are the rules of microbiome variation within a host species. And here we think about humans because there's so much publicly available data, and of course there's human health relevance to those types of questions. Now, if you take an overview of the human microbiome, of course, there's biogeography to the microbiome, different types of species in different locations. The cellular numbers, of course, 50% microbial and 50% human cells in our body. So we truly are a collective of bions, or a holobiont system. And within our guts, we have 500 to 1,000 bacterial species that comprise a lot of our ecosystem diversity in the microbiome. Many of us are aware that there's also a rich genetic diversity in our microbiome, 100 times more genes than in our human genome. And that genetic capacity is essentially sitting there either as a threat to us or as potentially an assister to things like immune system, digestion, metabolite, hormone synthesis, etc. One of the things that fascinates me is that our genomes are so identical, but yet our microbiomes can be quite variable. And so arguably, the biont and the genomic diversity of our microbiomes can help explain maybe why do some of us get sick to a certain threat or pathogen than others. Why are we better at digestion? Why are we better at various metabolism pathways? There's a whole suite of possibilities given that a lot of this diversity stems from our microbiome. And of course, a lot of this has been focused on diseases, where a very large number of diseases associate, maybe not causally, but statistically, with microbiome changes. And so this really sets up a big frontier ahead for the human microbiome and medical sciences. If you were to sort of turn the page in the textbook on the human microbiome sciences, I think that you would see some starting lessons. One is that geography influences human microbiome variation across the world. That is, where we live can influence which microbes we're exposed to. This biogeography and body site variation I just mentioned is a consistent factor in explaining microbiome variation. There's, of course, declines in microbiome diversity in westernized countries, which are also associated, though not causally so in a major way, with an increase in chronic illnesses in westernized countries. So there are some trend lines that we have. But, if you think about what else is underneath the hood here, what are the other rules of microbiome variation we're going to find out across humans? One of the things that we started to think about 10 years ago is 70 to 90% of human microbiome studies have been done in white European ancestry participants. It's really overlooked the diversity of all of us. And in fact, a study by Ron Bleckman's lab in PLOS Biology a year ago really highlighted how concentrated the biases are in human clinical cohort studies for microbiomes. In pink here over time is the European, North American kind of ancestry subject participants, and they dominate the world population that's been involved in clinical microbiome studies. You can see that starting to change over time. There's a lower relative abundance of this pink category here, to the rest of the world. So that's a step in the right direction, and exactly what we were thinking about, as well as we focused in our particular case on diversity in the United States. It's a single country, so that consolidates some of the global variation, although clearly there's a lot of variation within a country. But at the time we began our studies, we thought that we could start to analyze some patterns across two publicly available data sets, which were the American Gut data sets, a crowdsourcing campaign, and the NIH federally funded Human Microbiome Project. We wanted to do this with a lens on diversity, and we wanted to sort of see if there were any important trends that we could excavate that would highlight why studying further diversity in the microbiome sciences is indeed an imperative today. We are surprised that in some ways the microbiome sciences ended up in the same spot that personalized genomics did. Genomics has had this problem of concentrated clinical cohort studies in European ancestry white participants. The microbiome sciences is relatively new. We should have known better in some sense, but here we are. It's important to put a lens on it. So if you think about why do this, well, clearly we need to unbias human microbiome studies to capture more diversity. Clearly, this is relevant in the context of health disparities, in part because self-identity groups such as ethnicity and race do show variation in inflammatory bowel disease, colorectal cancer, heart disease, a lot of the chronic illnesses that we've seen a rise in increases in, at least in westernized countries. So this is not just about diversity, it's also about what potential options do we have for explaining health differences and disparities? Of course, when we think about health disparities in self-identity groups, there's a number of factors that could influence microbiome variation. Healthcare access, education access, socioeconomic status, exposure to pollutants and where we live. There is a lot of data that we need to have a better grasp on when we do our human clinical cohorts in order to be able to speak to the factors that will likely explain or underpin some of the human microbiome variation across the diversity of us. Unfortunately, a lot of the data we had in our initial data sets were from simply ethnicity and race, because those are required self-categories in the clinical cohort studies. So these are social constructs that I want to emphasize that correlate with determinants of health and have potentially biological consequences. I want to highlight this through an example of, if we take an individual's gut microbiome variation and we ask, well, how does a healthy person vary with a person who's sick? Of course, we recognize that there are many cases where microbiome changes can explain or at least associate with the disease status. But we don't often think about what's underneath the hood there. That, let's say, take, for example, colon cancer, where there's a 20% higher incidence and a 40% higher mortality in self-identity groups that are self-identified as Black versus white. And if there's microbiome variation between these two groups, it's possible that that microbiome variation is a predictor or a precursor of the types of chronic illnesses that we do see health differences and disparities in. Now, another important point is that disparities are modifiable differences. They're preventable differences in health outcomes. So if we learn a bit about baseline variation across the diversity of us, we could potentially also have recommendations, policies, and guidances to structure microbiomes in a way that they may be subjective to less risk for disease if the microbes are causal to those. So in effect, understanding the social constructs of self-identity, as well as how our lived lives influence microbiome variation, is an important force of health equity to potentially influence the future of these disparities and differences. So again, I want to emphasize that self-identity is a social construct, and terms such as ethnicity and race are actually such high-level coarse variables that groups have rightfully labeled these as ghost variables, racialized terms, because they don't reflect what's underneath the hood of all the associative and structural drivers that might really explain why we have microbiome variation and why we have health disparity variation across these social identity groups. So with that premise, and of course we need all this data in the future, I'll tell you what we can learn about so far from self-identity data. So this is adult 16S phylum level microbiome diversity across 4 self-identity groups here. The key point is that even at this high level, there's phylum level diversity of the 2 most common phyla, Bacteroidetes and Firmicutes, in the gut across both the American Gut and the NIH Human Microbiome Project. Moreover, if you dig a little deeper and you say, let's take these two data sets and ask, how does self-identity associate with interpersonal variation versus common other factors that are often reported in the news and that have influenced somewhat of the culture of human microbiome studies? So Andrew Book's, a former graduate student in the lab, has done this work. He's now at Stanford, where he compared how big of an effect does microbiome variation have on age, sex, that is male versus female, and BMI, and also race and ethnicity. And what he found is that race and ethnicity can explain more of the interpersonal variation since some of these more common themes that we read about in the literature or the news. And crucially, race and ethnicity is the only factor that replicated in both cohorts for the American Gut and the Human Microbiome Project. So BMI, sex, and age are significant in one, but only ethnicity and race are significant in both. And this was important to us because the American Gut and HMP are two wildly different microbiome data sets. They're collected from different age groups. HMP was MSTP or MD PhD students. They were sequenced at 2 sites in the country. The American Gut Project, of course, came from individuals who submitted all over the country. So we felt that if we could leverage this, we could actually find out the signal from the noise. And indeed, that helped us resolve whether this was a factor we should continue to think about. So if you drill down from phylum to more finer scale levels of taxonomy, one of the results that Andy ended up on was that there were 12 reoccurring genera or families that varied in abundance in exactly the same direction across the American Gut Project and the Human Microbiome Project. So these are 4 self-identity groups. You can see that the variation is consistent across each of them for the 2 studies. And ultimately, these 3 groups comprise actually what's known as the Christensen-Liliaceae, or 11 out of 12 of them comprise what's known as the Christensen-Liliaceae Consortium. These are groups that actually tend to rise and fall together in the gut microbiome, and 11 out of 12 of them have been deemed heritable in a population genetic sense that is influenced by human genetic variation, their abundances. And 7 out of 12 replicate heritability in at least 3 or more studies. So this suggested to us that not only we're finding some interesting consortia dynamics here for these abundance directions and consistencies, but also that they may be linked in some way to genetic variation in the human genome. Now, this isn't just sort of a statistical exercise to find out sort of the signal from the noise. Christensen-Liliaceae, as folks who study the gut microbiome know, is also obesity linked in both mouse studies experimentally and in human studies across cohorts. And when you parse the data out in terms of self-identity groups, you can see that there's some interesting trends that not all self-identity groups have this pattern of Christensen-Liliaceae being obesity or BMI linked. So when Christensen-Liliaceae is absent in the white unfilled boxes versus when it's present in the filled colored boxes, you can see that in general, Christensen-Liliaceae presence associates with a lower BMI for 3 out of the 4 social groups. So this indicates that when we don't sort of take a deeper dive into population diversity, we are potentially missing some of the confounding variables that go into lumping everybody together. Now, the consequences for this are obviously relevant for understanding how BMI variation arises and what it's explicitly linked with at a more personalized or population level. We can also use machine models to do distinguishability detection for microbiome pattern recognition. In other words, if we train the machines on gut microbiome variation across the American data sets and then allow it to pick out feature recognition and then predict whose microbiome that gut microbiome belongs to, and perhaps it might be mine, that would be something that would tell us that there's distinguishability within the microbiome data sets that could afford some amount of knowledge about these differences. Now, I'm going to show you data on machine learning. For those that aren't aware, essentially these presented on an axis of true positive rate against false positive rate. And when you have a straight line or diagonal line that goes through them, that just means that it's a random classifier. It can't distinguish one correct prediction from another, a false prediction. Now, if the patterns are above the random classifier, this will be considered a good, better, and optimal classifier where there's a straight line up and a straight line to the right. Now, what you see here is from Andy, Ron Bleckman, and his former postdoc, Sambois, when they were at the University of Minnesota, is that both for the American Gut Project and the Human Microbiome Project, across these 4 self-identity groups, you can see that the trends are above the random gray line. And in fact, if you sort of boil all this down to a single accuracy prediction number, about 70 to 80% accuracy, a microbiome machine learning model can predict which self-identity group these microbiomes belong to. So yes, there is distinguishability that can be determined at this statistical level as well. Now, and remember this number, 70 to 80%. That indicates that it's imperfect. There's a lot of microbiome variation still occurring that does not associate with ethnicity or race, but does associate with individuality. That tends to be the dominant factor. When we go to a different age group, so here these are adult, healthy populations. If we look at childhood, that is from 0 to 12 years of age, and we kind of redo this analysis with machine learning model on childhood microbiomes, we can find an even greater accuracy prediction, 90% or higher, for both race and ethnicity. So this tells me that in the simpler microbiomes of childhood may actually have a stronger signal of these self-identity social groups than we do in adulthood, that there could be a waning that essentially transpires over the course of a life of these microbes that are associating with self-identity. If you look at the differentially abundant microbes that associate with ethnicity or race in adults versus children, what we found really striking, this is work done by a former postdoc, Liz Malott, who's now at the Washington University in St. Louis as an assistant professor. What she found is that 50% of the distinguishable taxa in children are also occurring as distinguishable taxa in adults. So this suggests that some of these taxa stick around for a long time and remain distinguishable across the self-identity categories. Important to note that very few of these, if not none of them, are vaginally associated microbes. So these are microbes that are acquired outside of the birth transmission route and are thus probably found by or acquired by lived experiences of these children. And I'm going to speak more to that in the next couple slides with Liz's work. So first of all, Liz benchmarked that the common trend that childhood age structures with microbiome variation irrespective of self-identity groups. So here, Shannon alpha diversity is plotted across the age groups that we had access to in our, what was ultimately an 11 cohort, 11 different studies compiled into a meta-analysis. And of course, diversity is lower in the first 3 months than it is in the latter 3 months. We can see age variation on an ordination plot as well. Now, if you break apart the analyses that we've done on self-identity to each of these age groups, that's where we started to find some interesting patterns as well. So what Liz saw early in life, 0 to 3 months essentially, there was no statistically supported association with microbiome variation at this period of life. Now, one caveat here is this is a little bit smaller of a data set. There aren't as many access to samples here as there are in other stages, but take it for what it is, the signal is missing here, which again would be probably consistent with some of the observations that the microbes that are differentially abundant in children are not ones that tend to be vaginally associated or inherited. So no signal early in life, but after 3 months of age, that's when we start to get statistical significance and association values of microbiome variation with ethnicity or race. So it looks like lived experiences after 3 months of age, and there are many of them could influence this: going to childcare, eating more diverse foods, socializing more with other friend groups or family groups, could all lead to this emergence of variation after 3 months of age. And as you might expect, it persists after 1 year and beyond. What I'm showing here is 1 to 3 years of age, but it also occurs for 3 to 12 years of age, data not shown. These are, modest predictive values, right? So modest significant values and association values, I should say. So 1 to 2.5%. Sometimes it goes up to 5%. This tends to be where we see ethnicity and race falling out in sort of all of our human microbiome studies to date. So anywhere between, 0.5 to 5% variation is pretty normal and not outside the range for many other factors, as you saw for age, sex, and BMI. Okay, so at this point, the pattern is reoccurring from children to adults with down a specific taxa with various statistical tools. What we were then motivated by was what actually explains this. And in conversations with friends and colleagues across the desk, a lot of ideas would come down to two things. It's diet, because different Social groups have different diets that might drive this microbiome variation, or it might be something else that's physiological that might reflect structural determinants of health status, for example. So we tested the diet and geography aspects of this kind of pattern. And the way we did that was with a Nashville cohort of females. So we controlled the sex that were 18 to 40 years old, that were healthy, that had a healthy BMI, so not too low, not too high, and that all ate conventionally a Western diet, and they came from these two self-identity groups. They were not pregnant. They were not on a lot of medications, if any, except for hormonal contraceptive use, which we do have metadata on, and some prior antibiotic use that might have occurred 3 months or more before they entered the study. So our hypothesis was, if we take groups of individuals who have potentially different diets or different incoming microbiomes, at least, and we put them on the same exact diet, and we literally gave them the same exact food, controlled in a way in which their body weight was not expected to change. So the caloric intake was matched to body weight, a little bit more food for person X and a little less for person Y, but the same exact nutrient profiles in terms of macro and micronutrients. If diet has an effect, we might expect that microbiome variation comes in before the diet, and then it sort of collapses into something that's influenced by diet. It becomes more equal between the two identity groups. And then once they go off the diet, perhaps we start to restore some of that microbiome variation as they go back to their conventional habitual diets. Now, this was a dietary intervention that was done for 4 days with vegetarian intervention on Western diet folks. We monitored the microbiomes 2 days before and 2 to 3 days after the diet. This was an expensive study. I wish we could have had higher sample sizes, but to give everybody the same exact food, so they're not just a nutrient, but all the same types of vegetables, fruits, etc., turned out to be pretty costly. Now, if there was no dietary effect, the microbiome variation would persist across the identity groups, and we really wouldn't see any collapse of the variation. Because of the precedent that diet has set in the human microbiome field, I was ready to think that the dietary effect was going to be noticeable and detectable in this particular study. Now, the dietary intervention, 3 plant-based meals and 1 snack per day. Calorie content was personalized to maintain weight, as I mentioned, and they suffered through this dietary study because they had no caffeine, no chocolate, and no alcohol. So all the vices were eliminated, in part because we also measured metabolomes. So caffeine, you'd have a large spike in the metabolomes. And on occasion, we could see a caffeine spike, and that subject may have cheated and drunk a little bit of coffee. In our cases, we would remove and just double check that the analyses were consistent pre and post data removal. Okay, so we measured sites from the saliva and from the stool for microbiomes and viromes. So microbiomes for metagenomics and viromes for metagenomics. And we also took blood and urine samples where we did metabolomics on them. And I'm not going to talk about metabolomics today, but note that these metabolites in the blood and urine were pretty responsive to the dietary intervention, as one might expect. Okay, first of all, did the diet actually create a different source of nutrients for the subject? So what we're showing here are ordination analyses on the dietary macro and micronutrients. The habitual diet is shown in this bigger circle here in cohort 1 and in cohort 2. And then each day of the diet you can see is significantly different from the habitual diet. And often the dietary nutrients for one day were different from the dietary nutrients of another day. So there's overall variation to the habitual diet and then variation in the dietary profiles between the days. A little bit stronger for cohort 1 than cohort 2, but there was significance across both cohorts for the diets being different between habitual versus the study diet. Okay, good. So thumbs up on that. How is this going to affect microbiome variation? Well, the biggest signal in the data from both the fecal microbiomes and the saliva microbiomes or the oral microbiomes is that individuality rules the day. Something like 70 to 80% of interpersonal variation is explained by individuality. So you're seeing that here with individuals color-coded on these dendrograms of the microbiome beta diversity relationships. So hierarchical clustering relationships shown here. What this likely means is that the diet did move individuals' microbiome, but it didn't move it to a degree that it would affect some of the individual variation that we see. And that's emphasized here. So this is, I should mention, work done by Liz as well. Junhui Lee, who is a postdoc in the lab, is now at Cork University. and a former grad student, Rob, who's now at Estee Lauder working on skin microbiomes. What they have shown in this study is that diet has a really subtle effect on microbiome variation. So while significant, that is, it associates with before, during, and after variation, you can see how much overlap there is in the total microbiome variation here for cohort 1 and cohort 2. In contrast, the microbiome variation that associates with self-identity groups is consistently significant throughout, before, during, and after the diet. So it doesn't matter what stage we're sampling, and here we're showing all stages together for cohort 1 and cohort 2. There is a signal of microbiome variation that occurs recurrently throughout the study. Moreover, there are some taxa that frequently vary in abundance between self-identity groups, as we mentioned in the 16S analyses from the Human Microbiome and American Gut projects. And so we kind of re-looked at those and asked, were any of them differentially abundant? Do they include heritable taxa that we previously found? So there are a large number of taxa that have abundance variation between the groups. And in cohort 1, this includes 5 heritable taxa. And in cohort 2, this includes 3 heritable taxa. So a bit less than what we found before, but still some consistency there. And one of them is the Odorobacteriaceae, which is a highly heritable microbe, also a butyrate producer involved potentially in health or associated with healthy aging and cardiometabolic health, and it has differential abundance across the groups in our clinical cohort study. So it just shows the consistency of finding an ethnicity or race signal in the data. It doesn't matter whether it's a 16S study or a metagenomic study, and in some cases, there are particular taxa that hold a signal throughout all the analyses we've done. Now, because we did metagenomics on both the microbiome and the virome, that is the enriched virome from viral-like particles, Junhui, Liz, and Rob were able to analyze these associations with metagenomic functional categories. So conserved orthologous genes, antibiotic resistance genes, and then carbohydrate digestive enzymes or CASIMs. And it spans the gamut across the metagenome of the gut and oral cavities, and then also the gut virome on the bottom here, that a lot of things remain significantly associated with self-identity groups. COGS, ARGs, take, for example, the oral metagenome, there's a 13.2% R-squared value that associates with self-identity groups that can explain this kind of interpersonal variation. And really what takes the prize is the oral metagenome, because for CASIMs, there's a whopping 33% of the variation explained by these 2 self-identity groups. And what's interesting is that if you look at carbohydrate digestive enzymes, you can sort them into fiber breakdown or sugar breakdown. And indeed, there's a difference in which self-identity group has more fiber CASIMs versus sugar CASIMs. So this might be an indication of lifestyle differences that reflect the types of microbes that are in those 2 types of oral cavities. Now, to take a lot of data and simplify it and get to what I think of as the guts of the matter, what we're showing here is a heat map, and the stars are significant of the data for both metadata, so ethnicity, antibiotic use, and hormonal contraceptive use, across taxonomy and the metagenomic functional categories. And I'm going to simplify this by showing you where there's consistency. So the gut microbiome consistently associates with taxonomic variation for ethnicity or self-identity group, antibiotic use, former antibiotic use, and hormonal contraceptive use. On top of that, though, and what I'm personally struck by is that the gut virome holds a lot more significance for taxonomy, COGS, and ARGs across these 3 categories than the gut microbiome does. It's as if the virome is telling us, look over here, there's some pretty interesting signals that the microbiome can't quite capture yet. And for me, that sort of makes sense. If you study selfish elements, you know that they are the great diversifiers of the microbiome in our particular genomes. And of course, that's been observed early on, since this foundational paper in Nature in 2010 by Alejandro Reyes, where they essentially looked at infants and looked at fecal viromes and fecal microbiomes and asked, what's the metagenomic variation in the virome versus the bacterial metagenome, with color coding indicating the categories of functions, and each individual shown as a bar chart here. And the signal is very clear that microbiome metagenomics is highly conserved across individuals, but metagenomic variation of the virome is what distinguishes these metagenomes. It's as if they have the genetic diversity, the genetic signals, if you will, and the functional capacities to tell us where the variation inter-individually is and also across self-identity social groups. Now, the trend line points up for the use and study of ethnicity and race in the microbiome field. This is an excellent cautionary paper by DeWolf et al. That I recommend everybody read, and I'd already referred to in terms of the ghost variable issue. I think the goal here from this work and others is that eventually ethnicity and race need to be eliminated as ghost variables. We want to stop this curve from going up, and we want to have a plateau and come down as we actually get the metadata underpinning some of these associations that we're seeing. And I'll offer some advice for us as we go through the next few slides. So getting to this, set of why we study microbiome variation across the diversity of all of us, unbiased studies incorporate better metadata, understand what's happening and shaping health disparities and disease incidence differences, but also eventually in the long run, develop recommendations, policies, and strategies that might include more personalized or population therapeutics and diagnostics. Now, in that spirit, I also want to add one more piece of data that Rob finished up his PhD on, which is if you look at microbiome-linked human genes, that is, genes that control or at least associate with the abundance of a particular microbe, and ask, do they associate with diseases across biobanks and across ancestry groups, different ancestry groups? Rob found another interesting result in this story. What we did is we took 90,000 subjects in the BioVU medical records database, which is linked with genotyping data. We stratified those data by European and African ancestry and compiled a list of existing findings from the literature on 900 genes that had been previously reported to be microbiome linked. And then we took those genes in the database and asked, okay, you associate with the abundance of a microbe. Do you also associate with the disease? And what are the common findings among that? Now, in European ancestry, we're looking at these Manhattan plots that show us significance above the red dotted line. The particular genes that had an association with, let's say, the circulatory system. So think of the cardiovascular associations with the microbiome, or the endocrine metabolic system in green here, and so on. 31 associations among 10 genes in the European ancestry group. Those are shown physically on their chromosomes in terms of locations and the number of associations. Now, if you do the same analysis for African ancestry, you find no statistical significance that crosses the line. And what we're left thinking here is the biases in the databases could be creating some of the statistical differences here. That is, we don't have enough microbiome variation, we don't microbiome knowledge, we don't have enough genomic knowledge in the databases. And our large clinical cohorts that are focused on white European ancestry are creating a biased set of data that's giving us statistics in one group, but not in another group. So there's an emphasis here that we need more data, and we're clearly think we need more data through the lenses of the work like this FEWA study, but also our microbiome variation study. There's a whole swath of literature that is important for all of us in the microbiome sciences to digest and incorporate and grow from. Plenty of discussion on race, ethnicity, social variations associated in terms used with microbiome studies. And I would add into this that we are in desperate need of collaborating with social scientists in the microbiome sciences, because they will change the dialogue and discoveries for the better. And we at Penn State have formed a social biome working group where social scientists and microbiome scientists now talk to each other on a weekly or monthly basis throughout the year and submitting projects together, because the science is moving fast. And yet, this science is not just going to be about social and lived experiences, but environment differences, pollution differences. So we're going to need population health folks, geographers that are well positioned for this kind of work. So we need that. We need longitudinal data across life, because we're just looking at snapshots so far. And I know this is a theme that probably echoes a lot of what Nandita thinks important as well. Functional mechanistic results. So we need to know who people's contacts are, what the diets explicitly are, what the stress levels are, et cetera, that can be correlated to the metadata. better strategies to study these factors. One idea is just take one social group, take one large population social group, and you're going to have a lot of variation in context, diet, and stress, et cetera. And you can associate microbiome variation within one social group according to those metrics, rather than comparing them between social or self-identity groups. And also, like I have emphasized before, there's an aspect to this that if we're successful in linking microbiome variation to health consequences, that there could be social intervention strategies and policies to curb these microbiome disparity links, leading to more personalized approaches. Okay, so in terms of the trajectory of life, because we are seeing implications for thinking about this variation early in life and later in life, one of the areas that's relevant here is the DOHAD hypothesis, or the developmental origins of health and disease. It's an early critical window when microbiome composition has an outsized effect on short and long-term health. So in the first 3 months, a lot's changing. Microbiome is becoming more diverse. Lived experiences are becoming more diverse. Immune maturation and development are occurring, ultimately setting up a physiological system that may be predisposed to higher rates of disease risk, especially in adult life, after all of the training has taken place early in life, or most of the training is taken early in life. So an important context of why all this data matters across life, and also how the metadata will be important at many of these different lifetime periods. Okay, I'll finally end on a big picture theme, which is a lot about what drives my own personal research in our lab's research. What is, thinking about what is a host biologically made of, we often turn to thinking about genetics in the nuclear system of host species, animals and plants and humans. And we often think that that's sort of a different beast than the microbiome. And I'd like to raise attention a little bit on how we can think about this as more of a conceptual continuum between the nuclear genome and the microbiome, rather than disparate and separate beasts here. So the nuclear genome is largely vertically transmitted, of course, but it experiences recombination and mix-up from sexual reproduction. It's not a single genome being inherited asexually over long periods of time. It is a mixed genome. And I say that in the context of our microbiomes are quite mixed, as well as we have generations and even during our lived experiences and lives. And of course, the nuclear genome is made up of not just one type of gene, but all types of genes: meiotic drive and transposon, selfish genetic elements. You might consider these sometimes harmful genes, sex chromosomes that differentiate the sexes, and of course, the good old autosomal genes. Now, the conceptual continuum I'm trying to highlight here, and what we think a lot about, is that the microbiome is far more complex, but not necessarily discretely different. The microbiome is composed of different types of microbes that are both pathogenic, helpful, or harmless, much like the genome has these kinds of dynamics, just at different scales. The microbiome in some host species, and including humans, can be thought of as vertically transmitted, let's say breast milk introduction from mother to infant. And in other insects and plants, it's much more exquisite transovirally inherited microbes. And of course, there's also a lot of noise, environmentally acquired horizontal microbes that change up the microbiome. And conceptually, that can be equated to sexual reproduction and recombination that mixes up nuclear genomes each generation. So we put a whole big circle, if you will, around these two things, and think that there's a lot of rich conceptual paradigm work, and also maybe new ideas to stem from equating the mathematics, ecology, evolution, and functions of these two parts of what makes up a host, and really a whole biont system. Okay, so with that, I'll wrap up and say thank you very much for listening today. In the first part of the study, we talked about 16S and metagenomic variation emerging at 3 months of age and continuing through adulthood, particularly in American data sets. In our clinical cohort study, we find very little to subtle effects of diet on a short-term dietary intervention. We find that the microbiome and virome stories are slightly different, that the virome has an interest now in telling us about what types of metadata might be statistically associated with variation that the microbiome can't do for a number of different reasons. And finally, if you think about the human whole system, that is the genomics working together with the microbiome, in our Foehr study, we can find interesting examples where genes that control microbes also control the risk of disease. And sometimes those form a tripartite continuum where the gene changes the microbe abundance and the microbe abundance is linked with the disease. And so there really may be this tripartite feedback between the three entities. So thank you very much. This is a snapshot of some of the members of the One Health Microbiome Center from Penn State. I hope you recognize some of them, and some of them are your colleagues. Thank you much for listening, and I can pause here and take questions as they come up.
Thank you, Seth. And folks, please ask questions.
You could do so by unmuting yourself or by raising your hand and the emoji.
Well, I have a question while folks are getting their thoughts together. So you answered one, which was really fantastic. You were starting to, when you're looking at differences across ethnicities, my initial question was, well, how much of this is driven by diet or other lifestyle factors? And then you went into that, and that was really cool.
Yes.
And I thought that your perturbation study with the vegetarian diet was very interesting. Do you, and I guess because you alluded to it, maybe the answer is no, but do you have temporal data, and do you see kind of like a temporary blip in the change of the microbiome and then maybe a return back to the original state or a similar state?
That's a great question. So at least at the level of the three categories, before, during, and after, the diet doesn't explain, more than 1% of the variation in the microbiomes. So we can say that at least at the bulk level, the diet has a very minor effect. Now, I believe if we look harder, and maybe with some of the tools that you're developing, we could do kind of strain tracking that we haven't done before. We do have anywhere between 3 to 9 samples per person across the dietary intervention. It just depends on, when they gave us a sample and if they could. So there's the ability to kind of track fine scale variation. We are starting to poke at that at a different level. We're thinking about actually the virome a lot right now, and what's changing with the viruses, and then how do those reflect changes in the microbes. But we aren't totally focused on, is the diet changing microbes? And that's the only question we're asking. So I think, in short, we can do a little bit at a course level what you do so well, and maybe get at some of the finer scale variation about which microbes at what taxonomic level change the most. Thanks for the question.
Thank you.
Yeah.
There's a question in the chat.
Okay.
I'll read it out loud, or unless Taddeo wants to say it. Okay, so the question is, what are some of the ways in which projects are coordinated with social scientists?
Yeah, great question. So social scientists love thinking about geographic diversity, lived experience diversity, including family structures, stress levels, access to health system, et cetera. And they have ability to take zip codes or addresses and then tell you so much about how different areas have different access to many different factors of their social lives. And the idea that we could actually explain health outcomes by adding that social data and the microbiome data together may be a more enriched and likely model to explain how certain outcomes are different for different people. So just as we're excited about having a conventionalized lexicon about how to talk about sensitive self-identity terms with the social scientists who think that, who use self-identity terms without any apprehensions. They're like, of course we should use these terms. They reflect structural racism and what the kind of stuff they're interested in. The social scientists are learning about maybe a microbial influence they hadn't previously taken advantage of. So our current projects are thinking about breast cancer and microbiome changes across a diverse group of individuals, thinking about statewide or national initiatives to diversify clinical cohorts for microbiome samples, so that we aren't just talking about this, but we are potentially leading exemplars of diversifying the microbiome sciences, so that in a few years we can hopefully equilibrate some of these relative abundance charts of how one group has dominated the study so far. Thank you for the question.
We have a raised hand with Peter. Can you go ahead and unmute yourself?
Yeah, hi. My name is Ricky, and I'm in Nandita Garud's lab. We're all watching the lecture together. really fascinating. So I wanted to ask, given the complexity of dealing with human data, and the sort of the lack of experimental tractability, even though you're able to do, limited diet interventions.
Yeah.
Whether you think there's a role in, experimental models like mice or, other experimental models in studying these kinds of like social and dietary factors, right? Particularly the social factors, and whether how one would apply that kind of knowledge to, understanding these sort of more, just subtle human social, environmental, and genetic factors.
I love your question. Thank you. And hi to the group. To be honest, your question motivates me more than I can help answer your question. I think that's an interesting approach to take. Right now, I'm personally interested in, there's still so much more we can learn from human microbiome studies. I haven't thought a lot about my studies. Perhaps I can answer this with a study from Peter Turnbaugh's lab. So they were looking at different social self-identity groups, found microbiome variation as well, and then actually took the fecal microbiomes and gavaged them into mice, and then looked at the metabolic responses of those mice receiving those different microbiomes. And indeed, there were different metabolic responses and the kinds of things that might relate to the typical factors like studying BMI, weight gain, etc. So that's a really interesting way of going about sort of functional studies of these microbiome differences, a little bit different than the social aspects you're bringing up. Maybe we could do gavages with human microbiomes from different groups and then look at social activity levels and behaviors, which sounds like an excellent idea. So thank you for bringing that up. And I hope we can find some ways to do that, or other groups can, because that'll move us past the discovery and pattern seeking phase to something a bit more mechanistic. Was there another question?
I think we actually have another question from Michael Wozni in our group.
Hello, really, really cool talk. And I'm in the GRUD lab, and I'm very interested in, for example, the effect of diet on both species level and strain level kind of variation within and across hosts. But my question actually is about your exploration of the virome, and I was particularly interested, given that we see that across hosts, the microbiome is relatively conserved. Given that, do you guys have hypotheses as to why there is, relatively speaking, much higher variation across individuals in the virome, particularly given that, much of the virome is actually like phage or bacteriophage?
That's right. Yeah, a whole lot of double-stranded DNA phages here. And I think the answer to this is twofold. One is that phage genomes are the great diversifiers of bacterial genomes. So the first thing to change in a bacterial genome is usually the prophage insertion areas, because of high rates of recombination in phage genomes, as well as high rates of gene loss and gene gain, perhaps as that phage sits dormant and now the phages are losing genetic and therefore functional information. But that might go unmissed at a full metagenomic level, because the prophages might represent 1 to 5% of the genome size. And so there's all that other genetic capacity that kind of hides the signal if you're thinking about prophage-driven diversity. The second part is actually related to the fact that when we study the virome, we're looking at purified viral-like particle enrichments. And that means we're not looking at all prophages inserted. We're only looking at the active phages, the ones that have formed particles, likely to be lytic, although not necessarily true. They could just be kind of floating around in Brownian motion and not being very lytic. But what that would tell us then is not only are these phages genetically fastly evolving, but there are selective constraints on which viral-like particles have formed in the gut ecosystem, that your launching of viral-like particles or your production of viral-like particles is very different than mine and different than others. And so that would then say that perhaps some of that differentiation is coming from a confined set of viral-like particle activity rather than the total microbiome that has a normalized set of metagenomic variation. So I think those two forces are paramount and also offer really cool opportunities for further study and focus. As you might have heard, the NIH just put together the Call for the Human Virome program. I'm not sure if UCLA is participating in that, but if you are, then we're competing together, and all the more merits for us. I hope we end up together, because that is basically all these questions and fascinations really mean that the next phase of the Human Virome program will be a major boom to us getting a better handle on this.
Anybody else has a question, either raise your hand or enter in the chat, or just unmute yourself so we can call on you. All right. Looks like, let's see.
That might be it.
Yeah.
So let's thank Professor Bordenstein for his time, and really enjoyable talk. And thank you for coming.
Thank you.
We're going to stop the Zoom recording, and we're going to keep the link on for anybody who would like to chat with Professor Bordenstein to share a little bit about themselves and to just, get to know him over a virtual coffee chat. So I'll be here, and thank you again.